Okay, so we have gone over replication, where you take DNA, you start with DNA, and you make a copy of it, so you end with DNA. We have gone through transcription, where you start with that DNA and you transcribe it into mRNA. Both of these happen in your nucleus. Then your mRNA can leave nucleus through a nuclear pore, and it is going to go to either your rough endoplasmic reticulum or a free ribosome. Okay, and then we're going to start translation. Translation is when we're going to take that mRNA and we're going to transcribe it into a chain of amino acids. And that chain of amino acids is going to twist and form a protein. So we know that DNA codes for proteins. In order to get there, we have to go through mRNA. And we said that there are 3 kinds of RNA. So RNA, remember, has a nucleotide as its monomer. You have a phosphate, a sugar, and a nitrogen base. The nitrogen bases for RNA, remember, are A, U, C, and G. U is a T in DNA, but not in RNA. And we said that we have messenger RNA, and that is in your nucleus. And then it leaves and goes to a ribosome. You have ribosomal RNA that we're going to talk about today, and then you have tRNA. tRNA is transfer RNA, and it takes an amino acid and matches it to your mRNA. Now, in order for all of this to happen, your amino acids are coded by a DNA codon. A codon is a triplet code, so 3 nucleotides. So we're going to show how that works today. Okay, so we have our DNA strand here. Okay, so you have your DNA strand, and it's going to go through your helicase, and it's going to split like this, and it forms that replication bubble. And then your DNA polymerase comes and copies it, and you end up with twice as much DNA as you started with. Remember, DNA is double-stranded. Your A is always going to bind with that T. G is always going to bind with that C. Okay? And then your DNA goes through your RNA polymerase. Remember, that was the red one, and it binds, and then it forms a length of RNA. This is DNA. See how it's rounded? This is RNA. And remember, in RNA, we have a U instead of a T. So ATG, UGU, AGAG. Okay, and it's single-stranded. It's not double-stranded like our DNA. Now, inside here, we have our mRNA, which is coded through transcription in the nucleus, and then it's going to go out and find a ribosome. So here is our ribosome. Okay, a ribosome, it's purple. It's blue on the screen, but it's purple. Our ribosome is made up of ribosomal RNA. Okay, it has 2 subunits, a large subunit on top and a small subunit on the bottom. Okay, and our mRNA is going to come in here. Now we have 3 spots, if you can see them, an E, a P, and an A. Okay, so this is a ribosome, has ribosomal RNA and some protein. Then we have tRNA. tRNA look like this. Okay, at the bottom of tRNA, you have a code. Okay, each tRNA codes for a specific codon. But what's on here is the complement, so it's called an anticodon. So remember, we're looking for groups of 3 called a codon. Your tRNA has an anticodon, which is a complement that matches. That's the anticodon on one side, and on the other side, it's going to match with an amino acid. So you have 20 different amino acids. So this one matches with MET, okay? So it's going to attach up there. So what's going to happen is this is going to come through here. Your first one is AUG. So you're going to look at all of these tRNAs, okay, and find one that will match. Okay, now remember, we're looking for the complement. Okay, so if this is AUG, our complement is going to be UAC. It's the complement. So that's going to bind there. And then it's going to come here. We're going to bind the next one. It's UGU. So on our tRNA, we need to find one that is ACA. ACC. And when you find it, you're going to put it in there. Okay, now you're going to get a line of these, and they're going to come up. Okay, so that's going to move into that next block. And then your next tRNA is going to come and bind, and it has that amino acid on the top. So remember, when they move along here, they're going to look for a complement, okay, to the codon. So here's your codon, okay, and then you have your anticodon on your tRNA, and it's going to bind. Now, when they come up to here, your amino acid, right here, your amino acid is going to come off of the tRNA, and then it's going to be bound to the next one in line. Okay, so this is going to keep working its way through here, binding to the next amino acid. Your codon on your mRNA binds to your anticodon on your tRNA, and it's going to allow your amino acids to connect together at the top. And then at the end, as you're making your way through, you have a stop codon. On the stop codon tells your ribosome to stop and to release it. So now at the end, we have a ribosome. We have our mRNA strand, and when it comes out, your tRNA is going to leave and go pick up another amino acid to bind to the top. And then we have this strand of amino acids. Remember, you have 20 amino acids, and you have 4 nucleotides that can be arranged in a triplet codon. Okay, so some of these tRNAs are going to code for multiple amino acids, so the same anticodon can have the same amino acid. And then you're going to have multiple codons for most of your amino acids. This is an amino acid strand held together by peptide bonds. When it leaves the ribosome, it's going to then fold and conform into a specific structure and become a protein. Okay, right now it's just amino acids held together by peptide bonds, but when it leaves, it's going to form a specific shape in order to do what it's supposed to do, and then it will be a protein. Okay, so we've taken you from DNA to DNA and replication, from DNA to mRNA and transcription, and now from your mRNA into your amino acids, which form a protein in translation. Okay, go ahead and watch the other videos and answer the questions.